Hi,
Hey, Katarina. How are
how
you?
are you?
Great, thank you. How are you? What about you yourself?
Good, thank you. Everything good.
Perfect.
Are you Tomas or
Uh,
Thomas?
Either one is fine. Thomas is usually how I go.
Thomas. Okay. Are you from Germany?
Uh, Very close, guess. Yes, I'm from Austria, but like
Oh, nice.
5 minutes away from the border to Germany.
Oh, so close to Salzburg? Or
Oh, you know the region? Yes. That's the biggest city close by.
I love it. Yeah, I grew up in Germany, and I was a few times in Salzburg. I love it. It's so beautiful.
Oh, thanks. Oh, so where did you grow up then in Germany?
In the ugliest place, Bochum im Ruhrgebiet.
Uh,
Well, it's not so ugly anymore, but back then, when I was a child, it was still relatively ugly.
uh, Last time I forgot to try one experiment. Uh, do Bluetooth headphones usually work, or do they lower the audio quality?
Yeah, they work. Depends. If there are some noise canceling ones, are kind of sometimes you sound like people sound like they're in a tin box. But I think they figured that out. I think it improved a lot, so it should be fine.
Shall we risk it? Or
Yeah, you can try. We have a few minutes.
okay, thank you so much. Then,
Yeah, sure. Of course. Whatever you're more comfortable in is good.
then I will try the headphones just for the extended duration. But otherwise, it's also fine without headphones.
So, have you been following the European news about the Ukraine and the tanks?
Just a little bit. I only saw like the headlines. No, it's the headphones. Does this work, or is the
Yeah,
sound
it's a
great?
little bit lower and
Then let's do the other one.
okay. Yeah, but if you weigh more, it still works.
Sure.
It's still fine. We can still
Sure,
understand you.
perfect. Yeah, so many things happening in Europe.
Yeah,
Yeah.
but I'm so glad we are in science. I think always when the world is crazy, if you think about science work, it's fine.
Yes,
It's the only sane place.
yes, sir. Really love that. Like about like everyone actually trying to work together and like having kind of some shared things, even if sometimes disagreeing. It's different.
Yeah, it's at least we agree on like the principles.
Yeah.
Yeah. So I put up the link for the presentation, and then in the chat I added the paper, and I'm adding the lab website.
Oh, thank you so much.
I'm doing that now. And then I will start sharing on Twitter that we're about to start.
Hmm.
I'm explaining because I don't want to feel like that people feel like I don't care that they are around. It's just there are specific things you can really only do when you start the room.
Oh,
So I of course made poster, and so I make posters beforehand, but I feel like saying yeah now we're actually starting and everywhere and things like that is also important, and that you can only really do when it's actually starting. So that's good. Well, thank you so much for were you already on Clubhouse before?
no, just registered for this purpose.
Wow,
I will
thank you. That's an honor.
No, thanks for the invite. I think it sounds very interesting, this app.
Yeah, it started during COVID times, and I think it was the first audio only type of app, and then now a few other companies started copying it. There's Twitter Spaces, and I think LinkedIn and Facebook are trying something similar. But yeah,
How
I
do
think
you,
that's relaxing to
yeah.
not have a video.
What's the best strategy of discovering exciting houses and rooms?
So you can, when you sign up, find you can determine what interests you. So, and based on that, I think your hallway is put together. And so there are a few clubs that are really good. So if you like, it depends on what you would like to learn about, if it's news or specific news about specific topics. And then there are different clubs that are really good. So another good science club is quantum photonics. They have a lot of great physicists coming, and there is the Future Brain Club. That one is also, I think, really good. They also have then also like this news update rooms that are really good. So I guess the following the right people, it's then you get the right updates, I guess. But if you want recommendations, I can just give you some um, afterwards. And then there used to be tech news around the world. But they moved just to Twitter Spaces, so if you would like to listen to that, um, you it would be on Twitter Space, which is the same way. You just click on the room and then you can listen. Um, yeah, so I I think it's really interesting way of kind of making a podcast. Like I see it that way. So we then we record these rooms and then we also upload them then as a podcast on different platforms. So it's more interactive, I think, and more interesting than just having, if people come up and ask. Sometimes it's hard for people to have, like, there's a scientist with a PhD, and then some people are kind of not comfortable at asking questions. But I think by now we have more and more people feeling comfortable enough and participating. I think that's then very interesting when people do that.
Yeah, also for me that now promises to be fun with the questions. Looking forward to them. I think it's
Yeah,
really nice to also like see like also people asking questions from very different corners.
exactly. I think that is the most interesting part, and what got me basically hooked is that. And to be honest, since I'm doing this like club, before it was very informal before I created the club, like but I learned so much from science areas that usually you don't get to interact because you don't go to conferences out too much outside of your
Mhm.
area. So yeah, I think I learned a lot. So I think that's exciting. Yeah, I think in a couple of minutes we will start. Oh, Hamish is here. Hi, Hamish. He presented last week his work, which was really great. So I'm always happy when people come. Please feel free to come to the stage, everyone. We will start in a couple of minutes. And hi, Eli. How are you?
Doing okay. This looks really interesting. I unfortunately have to multitask due to the demands of my day, but um, definitely going to be uh, listening keenly.
Nice. Yeah, thank you. And hi, Hamish. How are you today? Hi, Deepak.
Hi, how you going? Yeah, good. I'm also
Hi, Kevin.
multitasking.
Hi.
Yeah, I'm looking forward to the talk, and I'll just be listening in.
Yeah, great. It's wonderful having you here.
Yeah,
Uh,
thanks.
everyone. And Hamish is in summer, so
Yeah, I'm currently just going for a walk, and it's yeah, it's going to be like 30. In Celsius is 30 degrees here, so it's already pretty sunny. Sorry, sorry to make you jealous, but I hope the weather's not too bad where you are.
yeah, it was raining and cold, but it's fine. It's fine. I hope you are enjoying it.
Yeah, I will. Yeah, thanks.
No. So yeah, I think we can slowly start. I know people will still be coming in, but we can start with introductions and then we'll go from there. So welcome everyone to Science Society, and of course a special welcome to Thomas here. And let me give you a short introduction so you get to know Thomas a little bit. Thomas did his master's in molecular biology at the University of Vienna, Austria, and then his PhD in systems biology at the University of Zurich in Switzerland. And he won different awards. For example, for best PhD thesis of the Faculty of Science at the University of Zurich, and the most tweeted bioarchive preprint, and which is really cool. And he also won the K99 Pathway for Independence Award, which is very competitive here in the US. And he thinks that the most intriguing part of biology hold the most promising avenues for research in human health and are still underexplored. So I really agree with that. And mitigating human aging will solve so many problems we have in health. So it's really an honor to have you here, Thomas. And before we start, we usually ask a few interview questions, if that's okay with you.
Oh, absolutely. And thank you so much for the invite. And thank you, everyone, for being here and joining us today.
Wonderful. So the first question is, how did you become interested in pursuing a life in science, basically? Was it something you always wanted to do since childhood, or is something else that kind of sparked your curiosity there? Thank you.
I think when I was very little, I wanted to become the first scientist who would discover living dinosaurs, but then that idea dropped off. And then when I got close to university, I wasn't sure what to study. And in Austria, there's this book that lists all of the different study programs. And one that was very appealing to me was a very science-focused study program. And I thought if I go into that direction, I will never get bored later in life. And since then, I kind of got stuck with it.
That is very cute. The story with the first living dinosaurs. I think that's for a lot of boys, especially, really a dream. My oldest son, he was a dinosaur fan too, so that is really cool.
But I presume that was before it was recognized that birds are dinosaurs.
That was before that, yes.
Well, but yeah, I'm glad you found, you got supported in following this path. And how did you then come into this specific research field of aging, and then also how this project came about? If you have if you could give us a peek behind the curtain there. Thank you.
Yeah, there are really like two main reasons. The first one, maybe the most important one, is people. Many of my mentors that I met during my postdoc were actually doing research related to aging or even collaborating on related themes. And even though I didn't start my postdoc in aging, it kind of gave me nice opportunities to learn more about aging and work with wonderful colleagues in different laboratories. And the second one is kind of more science-driven. So I'm really interested in the many things that we do not yet know about biology. And starting new research directions is something that occurs very rarely. One thing that I did was to look at all of the publications about biology that have been produced before and ask, where do people break into new directions? And presently there's 3 domains of science that are particularly open to novelty. One is research on diet, the second one is about eyesight, and the third one is about aging. So I thought this could be a good starting point.
Yeah, definitely. And I'm so glad because now we are here speaking with each other. I'm really glad you did that very systematically. I applaud you for that you checked. That's really interesting. I would recommend that for a lot of students, actually. It's a really good advice to look what actually also the fields that now fund like groundbreaking research, because I think there are fields that only fund very safe projects, and then there are fields where they tend to fund more projects like this that are more interesting. So
Absolutely.
congrats. And yeah, the presentation is pinned on top of the room. Please everyone access it while Thomas is speaking, and the stage is yours. Thank you.
No, thank you again, Katharina, and also thanks for organizing the full series, which is this large, wonderful volunteer activity that you're doing. Thank you for doing that. So thank you everyone also for being here today and being excited and curious about some of the research that we have been doing. In the next couple of around 30, 40-ish minutes, you will hear about some findings of ours about aging and how aging relates to unbalanced gene expression. On each slide, basically I will attempt to convey one main message. So if you have some question, don't hesitate to interrupt and also ask during the talk, but we will also have a discussion afterwards. The presentation that you will hear is kind of divided into 3 parts. At first, you will hear and see the background, learn what gene expression is, how that relates to aging. In the second main part, you will discover some of our findings, some of the new things that we have observed, and that will probably, hopefully, something that you will equally enjoy. And in the third, last part, we will zoom out a little bit to the bigger picture and make apparent and visible some of the things about the biology of aging that we do not yet understand. And with that, we can move to the second slide in the presentation deck, which at the same time is the most important one. So if you're here today, you're probably interested in science, possibly also in aging, and you might wonder why we age, what one could do about aging, if there could be ways also of mitigating some of the effects, and you will get some more insights during the presentation, and as you will hear about our own findings. But this slide really, I think, is the most important one. This is a slide from the United States National Institute on Aging, and this web page compiles many excellent guides about aging, things that we can do for healthier aging, for slowing down, also very practical things that we might face as we get older or as our dear ones get older, such as how to prevent falls. Falls are basically something that is rarely studied by scientists, but has this tremendous effect actually on our health as we get older. And it's really a really excellent collection of advice that I would highly recommend you to check later. So let's move to the science. On the next slide, you will see two different phases, and I want to acknowledge both of them. Both of them arrived independent of us at the same findings or very similar findings that you will see later. The first one is Cesar Payan Gomez, and he has been working as a bioinformatician in the group of Jan Hoymakers. And on the publication that you see highlighted on the right, Restricted Diet Delays Accelerated Aging and Genomic Stress in DNA Repair Deficient Mice, he actually also made the observation of how gene expression becomes unbalanced during aging. And the second person is Olga Ibanez Solé. Like ourselves, she basically found that this phenomenon that Cesar has also seen is actually a very widespread phenomenon that occurs in many different tissues in many different animals. She even was faster than me in figuring that out and also submitted to the very same journal. But the reason why you're not listening to her and instead listening to me is that I happen to be a little bit older, so I actually had submitted it before her. But she also independently arrived at the very same findings, and I really want to acknowledge that and also recommend you to check her preprint, which is presently on bioRxiv. So let's move to our manuscript of publication, which we published in December. You can see a lot of authors, and I want to thank all of them. So basically, a reason for having so many authors is that we actually inspected many different organ systems. And the main finding of our research publication is that aging is associated with a systemic length-associated transcriptome imbalance. So for everyone, especially those who I get to show something that they've never seen before, I want to introduce you what gene expression is and what transcriptome is. Basically, during gene expression, transcripts catalyze the production of gene-specific proteins. So next slide, you basically see these three entities. Genes give rise to transcripts, and they give rise to proteins. So in each cell of your body, there are roughly 20,000 protein-coding genes, and these genes template gene-specific transcript molecules that then contain that information of the gene and catalyze the formation of gene-specific proteins. And different genes, and thus ultimately different proteins in your body, they fulfill different purposes and thus are not always equally active at the same time. This is particularly prominent in some contexts. So if you go to the next slide, you will see these beautiful images of green and magenta on top of a black background. So what is it what you're looking at here? So these are basically fruit flies, specifically embryos of fruit flies. The magenta marks where the DNA sits, and green basically, this marks transcript molecules. And on each image you see the transcripts of another gene being colored. And by looking at these green signals, you're basically looking at these gene-specific transcript molecules. And without going into much detail, you can see that the green takes different patterns on different images. So sometimes it's in stripes, sometimes it's kind of in dots, sometimes these dots are evenly distributed, sometimes they're just like all together. You see that there's a lot of different things happening, and that's because different genes are active to different extents in different parts of animal bodies. And this is actually one thing that actually got me also very excited about studying transcripts as a way to understand how genes become active a long time ago. And I hope you equally enjoy that. So you can see that there can be strong differences in the activity of transcripts. But when we now move To the next slide, you will see a summary of something that not many people talk about. And that relates now to the biology of aging. Specifically, many research groups have reported that hundreds or even thousands of genes change their activity during aging. This means that they would find different transcripts to change during aging. You're here looking at 3 different cycles that basically indicate 3 different studies. And the number indicates the overlap between those studies and the number of genes that are found only by one study. And you can see that in published literature, studies done by very reputable labs rarely overlap, and they actually find different transcripts and different genes to change with aging. When we did our own pilot study, also under very controlled conditions, we would again see other genes to change during aging. Ultimately, there's only a single gene that is consistently changing in its transcripts across all different studies. And that could mean a few things. Regretfully, there might be a tendency to think that everyone else besides oneself makes things wrong, and obviously each lab thinks that everyone thinks that they are the ones that are doing things right and everyone else makes things wrong. But there could also be another reason. Maybe there's actually something much more fundamental to learn about the nature of aging. And the first hint towards this is presented on the next slide. When we repeated our own pilot study, we also found other transcripts to change. So this means that when we do the same experiment, again, under the same conditions, with the same people, with animals living at the same facility, and everything as identical as possible, we get different results. And that's very uncomfortable. Because that could raise a lot of questions. What's happening here? Why do different studies at the same laboratory find quite different genes to change during aging? Maybe the things that we observe during aging are not as general as we want them to be. Another explanation could be that we're really missing out on identifying the nature of the changes that are happening during aging. And a further hint for that came from a very enjoyable review that has been published around 4 years ago from Alessandro Celerino and Sandro Ori, which basically really asked the question, what have we learned on aging from omics studies? So omics, this is basically a set of experimental approaches that don't measure a single gene, but actually measure all the 20,000 genes, protein coding genes at the same time in one experiment. And they realized that in aging, many genes change the activity, so have different transcripts changing. But for each gene, the changes of the transcripts have a very small magnitude. So you also see here a summary graph of the results on human prefrontal cortex. On the x-axis, you see the magnitude of change. So if it is a log 2 of 1, it means that the number of transcripts either doubles or gets reduced to half. And on the y-axis, you see a summary of all of the genes that are found to change the transcripts with aging. And you can see that 95% of the transcripts that change with aging change less than 1-fold. So these are very small changes. You will maybe recall the images that you've seen before on the fruit flies. So these are basically on several orders of magnitude higher. In contrast, changes that are less than 1-fold basically are changes that a typical laboratory cannot reliably measure. So it's basically where we are in the domain of measurement noise. At the same time, this study they would find that many genes change. So let's summarize what we have till now. We know that many genes change in aging, but all genes that change in aging only do so to a very, very small extent. And you've also already seen that this is very different from kind of typical ways in which we think about regulation of genes, where you can even get these beautiful visible patterns that you can even detect by the eye. Most changes that happen during aging are too small to be reliably detected by themselves. So we took a step back and tried to figure out what might be characterizing the changes that happen during aging. And on the next slide, you will see a summary of our intellectual approach. So we basically considered mice throughout the entire lifespan, from young mice, 4 months old mice, to 24 months old mice. And our collaborators, they dissected different organs, basically trying to get a very broad overview about all different things that are happening in a mouse. In parallel, I went to many databases to sample as much information about genes and their regulation as possible. So genes can be, for instance, regulated by specific enzymes that basically bind regulatory regions close to the gene and then basically activate them or make them less active. And genes can also be regulated by sequences that affect how stable individual transcript molecules are. And genes can possibly also be regulated by other things. So overall, I tried to get as many possible explanations as possible, over 2,000 alternative properties or features of genes that could affect their regulation. And then we combined the experimental data with this potential for regulation. We did this technically through something called gradient boosting regression, which tries to pair those things in a very stable manner and allows us also then to ask what are the features or properties that contribute to this comparison. And we found that the most informative features would all relate to length, specifically how long the gene is, how long the transcript is, how long the protein is. All of those things actually also correlated. So we found that basically length alone would account for nearly everything that was reproducibly measurable. On the next slide, you will see a representative example. You see some cloud, and I will now walk you through the elements of the graph. On the x-axis, you have the log transcript length. So transcripts basically are polymers that consist of nucleic acids, and basically you see here you have the number of nucleic acids that are in these polymers, and you see on a logarithmic scale that it spans several orders of magnitude. On the y-axis, you have the relative fold change of old animals over young ones. So things that are above 0, these are transcripts that show relative fold increase with aging, and those below 0 show relative fold decrease. Looking at the visual, you also see that things go from top left to bottom right, which means that there is this strong negative correlation. The cloud is a density map that basically shows where all of the transcripts lie. So basically on the outer rim of this cloud, it contains 90% of all transcripts. And you also see some dots. These dots are the dots that would usually be detected by conventional approaches of measuring changes in transcripts. And you can see again that these changes are small. But what you also see is that although by themselves changes of the other transcripts are not reliably measurable, even in the cloud there is something going from top left to the bottom right, meaning that it contains information. So basically we have found that there's this effect that seems to affect most or all genes and leads to negative correlation with transcript length and age-dependent change of transcripts. We then asked whether this observation is specific to our dataset or if it might be something more general. And on the next slide, you will see a summary. So basically, we considered all of the popular studies that exist about changes of gene activity in aging. And so we looked at many studies of mice, many studies of rat, humans, killifish, everything we kind of could find. And we then asked in the published data and in our own data, how often are these correlations? And you see here on the very left, the share of tissues in human studies, which are the ones in gray, or in model organism studies, so this is everything except humans, that have negative correlation. And over 60% of all tissues in existing and data have a negative correlation. On the very right, you see a positive correlation. So we also find a few tissues which actually have a positive correlation in humans and in model organisms, which again is very interesting. And we believe that this negative and positive correlation are basically vis-a-vis manifestations of the same phenomenon. Only very few tissues do not have any correlation. So this shows that this thing that we have observed, this negative correlation between transcript length and age-dependent change, seems to be a very general property of aging. But there's even more to it. You might be wondering, so what happens now that transcripts change? Could there be anything that, any way that this could contribute to aging even further? And we actually believe so. And we have some strong support for this. Specifically, many people have already studied aging. And many things are known about aging. So we know about hundreds of genes that either extend or shorten the lifespan of aging, of animals. This still doesn't mean that we can fully understand aging, but it means that many things can contribute to the manifestations of aging. And we find that the shortest genes only rarely have genes that extend lifespan, whereas the longest transcripts, they belong to genes that would actually extend the lifespan. So we actually have this systemic effect on gene activity according to length, which basically unbalances the gene expression of cells such that genes that would extend the lifespan actually become less active as we age. For those of you who maybe have already studied some literature about aging or biology in general, I also have some more information on the next slide, where you see this red and this blue circles, and you see individual scientific terms in red and in blue. And the red ones, these are terms that relate to cellular processes that are enriched amongst the shortest transcripts and depleted amongst the longest. And the blue ones are cellular processes that are enriched amongst the longest transcripts and depleted from the shortest. And you see many things that you might have encountered as a biologist, such as chromatin or telomere organization, mitochondrial function, proteostasis, things that are inflammatory immune responses, many things that actually had been independently linked to aging before us. You also see that the longest genes are enriched for neural activities, which might help to explain some age-dependent neurological phenotypes. So overall, we see that there's a strong kind of organization of the genome according to length that overlaps with things that people have already been studying in the context of aging. And you've also seen that in most, but not all tissues, the longest ones become less active as we age. So what can we do about it? In the next slide, you see that it's possible to counter this imbalance that we have observed. On the left, you see a summary of some interventions that we have also included in our manuscript. So basically, there are interventions that are known to extend in animals the lifespan. You might have encountered some familiar ones, such as caloric restriction, which basically means having access to less calories, or partial reprogramming, where basically cells are put into a state that is kind of more embryonic in a way, or partially in that direction. But then on the very right hand, and that's not published yet, we actually have also been working on additional interventions, and we actually have an unpublished drug that counters this imbalance to an even stronger extent than any of the known anti-aging interventions that had already been described prior our work. So this means two things. First is that the observation that we made really relates to this kind of biological process that we colloquially refer to as aging. And the second one, that it is actually reversible, and we have some ways of developing interventions. With this, we are already at the intermediate summary, the main part about the biology of aging. You've seen that the observed changes in transcripts in aging are primarily measurement noise, at least I've hinted at it. Also that the changes that are not due to measurement noise are fully or nearly fully explained by transcript lengths alone. And the length-dependent imbalance disproportionately affects genes that affect lifespan, which leads us to this overall theme of aging being possibly due to unbalanced gene expression that stems from differences in the length of transcripts. With that, we would actually like to know where this imbalance really comes from. And we're not the only ones that are interested in the question. So I want to share with you one beautiful study from published last week in Nature Genetics that also again saw this length dependent change of transcripts with aging. And they figured out that if there is damage to the DNA, basically the machinery that creates these transcript molecules becomes kind of arrested, and it kind of creates almost like a traffic jam, where then additional machineries that would usually read along the gene can no longer pass by, leading to long transcripts becoming less active. And I personally believe that this is, for us humans, a large part of the explanation, and at least points very well into what's underlying this observation that we have. But you've also already seen that this can't be the full explanation. For instance, you've already seen that in some tissues, the longest transcripts actually show relative fold increase rather than a fold decrease. And we also have some additional insights that also already show why that can't be the full explanation. And I will share with you some of these insights. And I now hope that in the next 5 minutes you will bear with me as we make a small detour. And I promise you we'll come back to aging later. Basically, research into human genes follows very strong and identifiable patterns. So in this next slide, you basically see on the x-axis the observed a number of publications for each human protein coding gene in Medline. So for some genes, there are thousands of publications. For others, there's only a handful. And in the y-axis is the output of a machine learning model that I created that basically is able to predict how many publications there are on each human protein coding gene. And without jumping into the details, you see that most things are along the diagonal. This means that it is possible to quantitatively understand and forecast research on human protein coding genes. And similar as you've seen before, for a study on aging, we can also tease these models apart to actually understand where this pattern comes from. And I invite you to look at some of my earlier publications on the topic, where you see 4 highlighted on the right. So this could now be taken to different directions. One is you could feel tempted to do what others will also very soon do and kind of try to become very, very mainstream in your scientific research. But I think the far more exciting question is actually to ask, what are the things that are important but lie outside of the general course of biomedical research? And on the next slide, you see 3 examples. This is now from a recent study from us that we also published in December, that try to understand how often are people studying, scientists studying genes that are known to be important for different aspects of disease. So you have 3 examples. The first one is glioblastoma. And I selected this example because it's almost 20 years ago since some large initiative, the Cancer Genome Atlas, identified through unbiased data genes that very strongly contribute to glioblastoma. 27% of those genes have never been mentioned in the title or abstract of any publication on glioblastoma. In the middle, you see something related to aging, Alzheimer's disease. Specifically, there has recently been a large $250 million initiative of the National Institutes of Health of the United States towards Alzheimer's. This initiative, they identified the most promising therapeutic targets for Alzheimer's disease, which is a devastating disease. And although many people study Alzheimer's, 44% of the genes that are the most promising therapeutic targets, according to the best available data, have never been studied, appeared in the title of any single or title abstract of any single publication on Alzheimer's. And when we move to SARS-CoV-2, again, there's much research happening on SARS-CoV-2. But if we ask which of the human proteins that interact with SARS-CoV-2 virus actually have been studied in the SARS-CoV-2 literature, 60% have never appeared in the title abstract of any publication on SARS-CoV-2. And I could go on and show that for many different types of diseases. So basically, there is historically acquired patterns of scholarship that still basically are propagated and lead to many genes not being studied. Imagine that you want to understand what a plane is, but you're only given half of the parts. It would be very difficult for you to understand what a plane is, even more to control it or to fly it to some other destination. And that's probably an oversimplification, because we biologists are still trying to figure out the principles behind biological matters, whereas we would be familiar with engineering principles behind a plane. And the same thing also happens with aging. That's what you now see on the next slide, and that's the last and second most important slide. And it contains a lot of information, and we'll walk you through it. Basically, that's actually the other way of how we ended up at looking at or finding this length association. We identified roughly 100 genes for which there's a very strong support that they relate to aging. And then we went to the literature and asked how many of these genes actually have been studied in the context of aging. And 64% of those genes, we know what they're doing in aging. And it's actually very reassuring that these genes that seem important, we're studying many of them. But then there are these other genes that have not been characterized well in aging, although there would be this large support that those genes associate with aging if we look at unbiased data sets. And we can also break this down into individual categories, and the specifics are not too important, but basically the higher up in the list, the easier it will be for designing experiments. And we can, in the middle, you see that we can gain additional information about those genes. Like in which organs would it be easy to studying them? And we can create here in blue different scores that tell us how easy it would be to approach these genes with different investigative approaches. We can also look at those genes in the general scientific literature and ask if those genes had been studied elsewhere. So one of the messages of this slide is that there are many more things that should be studied in aging, but no single scientist has ever studied what these genes are doing in aging, although there's a very strong, overwhelming support that they're doing something with aging. And Katharina in the beginning mentioned this K99 award, and which will allow me then this year also like to start a laboratory that will actually focus on systematically exploring all of these different genes that no one studies, although they seem to contribute to aging. And now let's jump to the very right-hand side, which is like concluding the full presentation. This is the proof of visibility. I cannot yet share with you the specific identity of this gene that I call here gene E, but we realized in the literature that gene E is required for the transcription of long genes. So we noticed that this gene also becomes less active in aging, and this led us to first look at gene and transcript length. And I think it's actually this gene which is the initial trigger for this length association in mice and also in some tissues in humans. And with that, I just want to thank you all again for your attention and also share with you a little bit the gift of this gene E. Take a second, look at the screen, realize that there is something like gene E. First time I couldn't believe it. I started to shake. It's kind of seeing all of many images flashing in front of my head, almost like picture yourself seeing all of these memories, like of young, old people, seeing all of the things that are happening around you, things that happen in each of your cells, and picture yourself feeling completely overwhelmed that you have possibly seen what drives all the deaths and all of the change around you, and just shaking. And I was kind of speechless. I couldn't really move much from the chair, even started to cry, kind of completely overtaken by realizing that by looking at this gene E, you might be among the first people to realize what's causing so much change in the life around you and many generations before you. And with that, I want also to extend the gratitude for you being here and joining this presentation. I also want to thank you in the name of my collaborators and all of the mentors. Scott Budinger, he has been in starting the study on mice and created initial data set. Alexander Sasha Misharin, he did many of the initial experiments. Rogan Grant helped with bioinformatics. Luis Amaral is also a fantastic mentor also on research about the research. Rick Morimoto has been a mentor and also a course on aging. Also very great career advice as Bess McNally, who's another member of my mentoring committee in the K99, and Jakob Snyder, who is like an informal mentor but has also been very instrumental in us navigating the path towards this publication. And I also want to thank funding, which includes the Northwestern Data Science Initiative, National Science Foundation Science Policy Grant, and a K99 Postdoctoral Tenure Track Award of the National Institute on Aging. And finally, on the last slide, you see some conversation pieces. I'm looking forward to the discussion. Thank you so much.
Yeah, thank you so much, Thomas. This is a brilliant work, and I really appreciate you sharing this with us and coming here, because I really wanted people to appreciate your way of approaching things in general. I think we have too little of that, looking systematic, to take a step back first, and to take the time to take a step back and look at it from a larger perspective, systematic way. Wait, how are we doing things? Why are we not making the progress we wanted to make? And then analyze large sets of data and then start from there. I don't think many people do this, and I wish more people in mental health research, hopefully people are listening, please take Thomas' approach, because that way we will solve so many things in a much effective way, and we save money, right? So I was very impressed with your work in general and how you're approaching. And then when I interviewed you and you said you approached the same, already looking for what in what field you want to go in. So I guess it's something at some point you learned as a person to analyze everything in life that way, and then you use that for your research. So it's really elegant and very important. So everyone, look through Thomas. If you're deciding in what career to choose, what field to choose, look at his publications where he goes through large amount of data, and then go from there and write your grants based on what is not published yet. So, and what would be very effective to do. So you did just that was a huge service, I think, for the field and for humans. So thank you for doing that.
No, thank you for the enthusiasm. And again, I should also say some of the most pleasant conversations I had and a strong level of support is actually also from funding agencies. I think like people that are like more leaning towards science policy, they're particularly, I think, also open and appreciative of some of this approach that tries to add something where no one adds more knowledge.
Yeah, exactly. And I want to open it up for questions. So please, Hamish, go ahead. Thank you.
Hi, yeah, thanks. Can you hear me all right?
Yep.
Yes,
Yeah.
thanks so much.
Yeah, thanks for your great talk. I was actually going to say a very similar thing to Katarina. It's very clear that you have gone about it systematically, and now you've found gene E, and it's very exciting. So I look forward to your publications to see what gene E does. So good luck with that. I might have to set up a Google Scholar alert to make sure I follow you.
Thank you.
But before you got to gene E, I was going to ask if you think that it's if there's some transcriptional regulator that's causing the changes in just the lengthy genes expression. And so I guess then the question is, well, what's controlling gene E? Because it sounds like you said that gene E expression goes down in aging. And I guess just maybe you don't know that yet, but I mean that was obviously interesting. And it tied into that because I was just thinking as you were talking. I mean, I'm not an expert on aging, but I understand that mammals, mammalian ages are kind of somehow set or adapted, and it might be to do with like child rearing and evolution. So humans live for a certain age, elephants and all that. So do you think that like say when gene E starts to decline, it could be the set point of when humans should start to die? I'd love to hear your thoughts on that.
I'm not so much sure about like if it's a static timer. I think many things relate to environmental exposures, and that there are things that integrate over the passage of time and lifespan. Yep. Sorry. Yep. Hey, sorry. No.
Oh, sorry. No.
I think that makes no, it was me. Yeah. So we know that in terms of gene regulation, gene E also is one of the most distinctively regulated genes that exist in humans. That actually has been shown in even like a separate branch of scientific literature. So it has a very specific way in which it is regulated. It's very different from typical genes. So I think that this probably relates to the regulation of gene E during aging. Then there's the evolutionary aspect, and that they were not yet sure what to think about. So like you've seen like on the genes that are short, and I only gave a very brief teaser on that, these tend to be genes that are involved in fighting pathogens, like inflammatory genes. And short genes, because it takes time to kind of transcribe a gene to transcripts, they can be actually produce proteins within a couple of minutes, whereas long ones, it would take several hours to produce something which might be too slow for the response to pathogens. So I think it could be that there's actually a trade-off, at least for short genes, that several of them are more responsive genes that help short-term survival, but might do so at the expense of long-term survival.
Right, yeah, thanks. That's really interesting. Yeah, all right. Well, thanks for your talk and look forward to seeing your research.
Thank you.
So this hopefully will be an easy question. There was the slide that showed that most genes had a negative correlation, some had a positive correlation, and then there were the smallest category was tissues with no correlation. I'm wondering, the tissues with no correlation, do those perhaps tend to be tissues that show less signs of aging?
Oh, that's an excellent question. I can say that some of those that show the signs of aging the earliest are also those that have the strongest negative correlation. So those in the middle, I will have a closer look at them again. Actually, it's a good idea. It could be like that there's like some multimodal thing where like either extreme or middle kind of behaves different from the rest. But overall, they don't stand out for aging faster.
Look forward to
Yes.
to any findings.
Yes, no, it's excellent. Thank you so much for. It would be really interesting actually to see how they actually change physiologically. These ones, we didn't look at them too much regretfully yet, but there could be an very interesting story there. And also like to see maybe these are tissues that respond less to environmental stress and other changes of gene expression. So that would, yeah. I must definitely have a closer look at them again. Thanks so much for suggesting that excellent point.
Dr. Shah, do you have a question, or Deepak?
Yes, thank you so much, Thomas, for your wonderful talk. And my question, as a part of the slides, I noticed you put the FGF21 gene, and I was just wondering about your observation about this gene specifically, because we know about the important role of that, especially in relation with the insulin kind of, for example, IGF and all of those insulin related component. Also, in a case of the cytokines, I was wondering about your observation and how it might be used for the future, for example, gene therapy. Do you see any possibility or not? Thank you.
Yes. So basically one interesting thing about this factor is that it's kind of secreted and could possibly circulate. So I think that opens the very interesting question of kind of which effects happen close by to the sites where FGF21 would be produced versus somewhere else. So I think from that potential, it is very interesting. It also is in when we went back to our old data sets, we found it actually also to have actually the most pronounced effect amongst the known anti-aging interventions, which could indicate that these are the things that happen below it are the more important ones, which could tie to as indicated, maybe something with IGF, but it could also mean that maybe it has a better way of spreading to many different locations, or kind of affecting things kind of at more sites, which would be very interesting. There are also some human mutations that also affect FGF signaling, and these seem to be mutations that associate with exceptionally long-lived humans. So it could actually also be something that might be physiologically really relevant to humans. But I don't know yet exactly how it directly links to the observation that we made here on length, and whether it's like a more immediate or less immediate effect. But it definitely seems very promising as a general, also therapeutical direction.
And yeah, so because we just talked about the imbalance in the expression, also this gene can be related to imbalances in the metabolism. So that's why I
Yes.
just thought that I asked you about it. So thank you so much. I'm passing the mic to the next person.
Yeah, Amir, go ahead. Thank you.
Yeah, hi everybody, and thank you, Thomas, for the excellent presentation and the wonderful work that you have done. Congrats! And by the way, I was excited to hear that you studied in University of Zurich. We might have shared the table in the Mensa or in the library, because I was studying in ETH Zurich, and still it seems we graduated almost at the same time. So yeah, I was thinking of the first thing which popped into my mind when you were talking about the length of the transcripts was that the phenomenon of alternative splicing. And as you know, every human gene has a couple of in average I don't know 3 or 4 alternative isoforms, and some isoforms can have significant differences in length. And I was wondering what your level of analysis was. Did you, calculate the average length of all transcripts of a gene, or actually every dot on your scatter dot plot is a single isoform, not a gene? So you did the analysis at isoform level, not at gene level.
We did it on a per gene level, mostly because the initial experimental data we thought would not provide sufficient information for per transcript resolution, particularly like when it's more experimental data to reliably distinguish different transcript isoforms. We think that it could relate to. So I know from the structural properties, and I skipped over that. We also considered properties of the number of different transcript variants, number of exons, and so we considered those as potential things that could contribute to our regression model, but we did not find these things as informative. But now going back to splicing, there also has recently been, I think, a preprint, I think it's from Vadim Gadishev's lab that showed that in humans, in the same data set that we actually investigated, there is actually changes in splicing that are happening during aging, and they concluded that that one of the things that breaks in aging or changes in aging is actually the splicing. One thing that is, I think, very interesting is so there's this physiological thing that has often been put in the proximity of aging, which is heat shock response. So basically, if you administrate heat, then some proteins will kind of unfold, similar to like egg white, kind of getting white if you cooks it. And this heat, and there are certain things, proteins that should kind of counteract this from happening. So like that maybe makes things proteins not stick together. And basically, one of the things that's happening in heat shock in human cells, and that has been published last year, I think by Kugusi et al. In Molecular Cell, is that the machinery that transcribes genes to produce transcript molecules runs faster. And because that machinery runs faster, so there certain parts of introns cannot be covered in time by other proteins. And because these other proteins can then not cover these parts of the introns, what's happening is that the transcripts that are being produced because the machinery runs faster, they expose certain stop signals that are in these introns. And that in a way is different splicing too, but it also is this alternative polyadenylation, polyadenylation being this kind of signal that says when to stop transcripts. So what's actually happening is that there, because the machinery runs too fast, you have a certain type of distinct splicing that then leads to transcripts being less stable. And this also leads to a length-associated phenotype. So I think it can be related to splicing, but I don't think that actually splicing is the only thing. But yes, absolutely. So I think there is something with splicing, and it probably also relates to why splicing might differ in aging, and it could be inherently tied to producing transcripts that are less stable.
Yeah, follow up on that, not related to splicing. We had last week, or over the last 10 days, two speakers that talked about the 3D structure basically of the DNA. And one was Dr. Bazou. He is at Durham University, and he basically managed to make a predictive model of the genetic code and how the 3D structure will be, how the folding most likely will be of that gene. And then the other was Dr. Marie Nigambor. She's in Barcelona, and she did this MIOS imaging to see, like, to visualize gene folding at the nucleosome resolution. And I was thinking, is it just more likely for a longer gene to, over time, that the 3D structure is just more affected than in a shorter gene? Because just, we have G4s, IMs, and I motifs and so on. And is it just statistically more likely for these to happen in aging and longer genes, maybe?
Oh yeah. So we haven't looked at it by ourselves, but from colleagues that basically study basically I don't want to spoil too much of their work because I think it's not published, but there is reason to believe that that long genes are at different locations within the 3D structure of the DNA. And some of these things also change during aging. We don't know if that just happens to be a coincidence or if it relates to the same phenomenon that we observe. It might just be like another side of the same observation, or if it is something different. But definitely the long genes seem different in the 3D localization. And I don't want to spoil too much because I think I'm not sure, because that's from a colleague.
Oh, that's
Yes.
exciting. Maybe he can come once he published.
Not from our lab, but like someone else outside. No collaboration.
Yeah, maybe we can invite him
Yes.
once he's published. Yeah, that would be great. I'm looking forward to that work, then. That's really exciting. And to your work that it's not published yet, and you mentioned, so it's really exciting. I know that Deepak, you were waiting patiently. Do you have a question still? Oh, maybe he he is away from the phone right now. Let me check in the chat. So we've been going over an hour already, so I wanted to check with you how many questions are still okay to ask, because you must be very busy also. So,
How many questions do we have left?
I think probably one or two, but
That's fine.
that's
1 or 2 is fine.
that's yeah. And then another thing that I wanted to ask about. It's so interesting, the length, that the length is so important. So we have the 3D structure, the splicing, which maybe, another thing that contributes a lot to these structural changes is pH levels and magnesium levels and so on. Is that something that you observe in like aging cells, that these type of iron distribution and also pH level changes happen more frequently?
Oh, that's an excellent question again. Yeah, so, you've seen me, you heard me talking about the transcriptome quite a bit today, but we were also wondering what is actually behind the changes that happen to proteins as we age. And quite a bit of that can be explained by the changes that we observe in the transcripts. But there are even more things that contribute to the changes in proteins during aging. And one of the things that we have seen to be very informative on this is actually pH. So basically, individual proteins, they basically constitute of these different amino acids, of which some like have a more positive or negative charge. And depending on this charge, they will have a different isoelectric point. And we see that according to the isoelectric point, this is actually one of the things that strongly informs on age-dependent changes in proteins that are not, and it informs on the changes that are not explained by transcripts. So we have actually been encountering that against more unbiased investigation. But for transcripts, we presently don't have strong support to link it to pH, but for proteins, we definitely have some implications.
Yeah, I was interested for a while in IMOTs and G4 related to synaptic plasticity in neurons, but I never got actually funding for this. I sent out a few times a grant, but yeah, and because a paper came out that showed in live cells that in eye motifs like pH levels and magnesium levels and so on could, increase the occurrence quite significantly. So that's why I was thinking of that paper. Then one more question was
Just before you
that
go on, I just wanted to interject that something like 40% of the U.S. population is magnesium insufficient, right? So there are two questions: what disease burden or morbidity burden does that contribute to? But at the same time, are your controls really appropriate?
Well, I think the connection just dropped. Do you hear me?
Yeah, I can hear you. I think Eli said, but I couldn't fully understand. So you mean how pH levels then in real life actually would affect aging?
Oh, I was referring more to magnesium.
I already wrote it down. It's a very interesting thing to test. We haven't looked at it, but it would be interesting to actually ask. This,
Yeah, I can send you the paper that I'm
yeah.
referring. I would have to look it up. I don't know it because it's years ago. And then the other question that I had is, are these long genes more like, is there a higher percentage of being highly conserved, or are these genes that are like newer, like
Mhm.
more mammals, or maybe even primate related genes?
So these are genes that are generally more specific to vertebrates. So like the oldest genes, they also tend to be relatively short genes. So many kind of housekeeping kind of functions or general functions of what cells do tend to be short. And when we then move towards longer gene lengths, it's genes also that change a lot during development or relate to tissue-specific functions. So these are newer genes. Yeah, absolutely.
Yeah, that's interesting. And then is there evidence? I know that beginning of last year, Dr. Detlef Weigel and colleagues, they showed that in plants, some highly conserved or important genes are very protected, even when there is replication of the cell going on. That there are specific, that basically mutation is not by chance, that you have regions that are highly protected. So are these, since it's newer genes and non-vertebrate, you have some that really basically very easily rejuvenate invertebrates. So are these genes maybe less protected because they are, not directly survival, but like aging is a luxury type of genes?
So one could twist that either way, the answer. So one is this one thing is that by being long, there's like more chance that something bad happens. Like there's like a higher chance that, for instance, mutations happen. So like long genes in a way are kind of predisposed to for the chance of something bad happening. At the same time, long genes have fewer kind of mutations than would be expected by their length, which indicates that at the same time there's like some protective thing going on that allows these genes to be long and still be okay or do something. There are kind of these seemingly opposite ways of looking at it, which are true at the same time. One thing that we found is that the genes that are very long at the same time are genes that would actually have kind of a good effect for survival or lifespan, which begs for the question of why they are long and how this relates to aging and evolution. And we don't have a strong proof for that, but I think there's like one hypothesis that I really like. So like the regulation of genes in yeast and in other in animals, like everything that's kind of more complex than bacteria, is awfully complicated. So bacteria seem to be doing just fine for most purposes. And one idea or hypothesis, I think that was like published as a kind of more perspective in Cell like around 10 years ago, is this frustrated gene hypothesis. And the idea is that gene regulation is not so much there to regulate genes, but to make it really difficult to control genes, and so that things should be there so to barely survive. And the idea is that that is parasitic nucleic acids, like basically you could think of them as certain types of viruses or so, that basically try like to propagate in the DNA, but are for organisms not nice if they're doing it. So the idea is that if the gene expression machinery is barely functional, if those parasitic nucleic acids would become active, the whole gene expression machinery will basically break. And basically that could mean that then the organisms die, which on the population level could be a way of restricting the spread of parasitic nucleic acids. And the way I'm thinking about kind of survival genes being long, or long genes extending lifespan, could be along these lines. I think that basically having these genes as long, there could be, if there's like unspecific types of damages to the gene expression machinery or these parasitic nucleic acids, that then there would be a reduced survival of the individual, which, however, on the population level might actually increase survival. So it could be that we might want to shorten lifespan of individuals to kind of prevent the spread of parasitic nucleic acids. But the last part is just extrapolation from my side, but it's a very good puzzle of why these genes are wrong.
Interesting. It's really fascinating. So thank you so much for this really amazing discussion and sharing your work with us. And we wish you all the best for the future, although I don't think you need any of our wishes. But yeah, and we are very curious to continue to follow your work. So thank you so much, Thomas. And it was really a pleasure
Thank
having
you,
you.
and thank you, everyone. Thank you. Bye.
Okay, and thanks everyone for coming, asking questions. It's so much more interesting to have a discussion with, with more people with different ways of thinking. So thank you for that, and yeah, I hope I hear you all back soon again. Thank you.
Bye.
Okay, I'll close the room now. In 3, 2, 1. Bye, everyone. Thank you.